Okay, so far we've covered the prokaryotic or bacterial microbes, and we've covered the eukaryotic microbes or the protistans, most of which were protozoans. Now we have one more group so that we have a full spectrum of microorganisms that we'll be studying within this course. These are the viruses. So now we'll begin to take a look at the viruses, which are acellular microbes. The techniques that we use for viewing and cultivating other microbes, such as the protists and the bacteria, are useless for viruses because they're much smaller than could be seen with the first microscopes. Now we have some very powerful microscopes that allows us to see them a little more clearly. Smallpox and polio were examples of disease that were known to pass from person to person, but for years the cause was really unknown. It wasn't until the 1890s that we got any insight at all. Louis Pasteur was the first that proposed the term virus, which is simply Latin for poison. In 1884, Louis Pasteur also designed the first vaccine for rabies. In the 1890s, there were substantial revelations about viruses. Ivanovsky and Beijerinck showed that a disease in tobacco was caused by a virus called the tobacco mosaic virus. And then Loeffler and Frosch discovered an animal virus that causes foot and mouth disease in cattle. They identified that an infectious filtrate passes through porcelain filter but remains infectious. This showed that infection could be caused by cell-free fluid, so agents that are much smaller than the bacteria that this filter was designed to stop. Many years of experimentation and research showed that viruses were non-cellular, that had a defined size, shape, and chemical composition. And by the 1950s, virology was a discipline of its own. Since we've known of viruses, there's been some argument about whether they're living or not. We might ask the question, where do they belong in the biological spectrum if they're not living? Well, they can infect every single cell type. That is, bacteria, fungi, algae, protozoans, as well as plants and animals. They're found literally everywhere in so many ecosystems. We do know that viruses cannot exist independently from the host cell, so most scientists consider that they are not living things. However, since they can direct life processes, they're often considered to be much more than lifeless molecules. They are technically referred to as infectious particles that are either active or inactive. Either way, they cause disease, so the argument of whether they're alive or not is almost irrelevant. We always consider that viruses have negative impacts. However, viruses have many positive uses also. They've probably been around for billions of years, although we don't know exactly how long. They have a very important role in evolution because they interact with the genetic material of host cells. So a virus can carry genes from one host to another. This process is called transduction, and we'll learn a lot more about it later. When we see that even in the average ocean water, there's 10 million viruses per milliliter, we can start to understand that they must have a significant role in the functioning of ecosystems. We've even seen as many as 250 million per milliliter in some lake water samples. Their role in evolution has really shaped the way organisms have evolved. For example, scientists are now beginning to understand that there's a lot of viral DNA in the human genome. Anywhere from 35 to 90% of the human genome could have originated from viral DNA. That's huge. When we think of the impacts of this, we have a whole field called epigenetics, which is about how chunks of DNA have moved around through the genomes of species throughout time. It's a fascinating field, but way beyond the context of this course. So viruses are obligate intercellular parasites. That is, they cannot reproduce on their own. They have none of the cellular machinery to make proteins, and thus they possess no enzymes of their own. So they depend on the host cell's machinery in order to replicate themselves. Viruses invade very specific hosts and then instruct its genetic and metabolic machinery to make new viruses and release them into the surrounding environment. This table summarizes some of the properties of viruses that we're interested in. I'll highlight a few here. First, they're obligate intracellular parasites. They've had a major impact on the development of biological life in the sense of their impact on evolution. They're tiny, ranging from 20 nanometers to 400 nanometers in diameter. They are not cells and have very compact economical structure. Independently, they don't fulfill the characteristics of life, so we consider them as non-living microbes. When they're outside the host cell, they're inactive macromolecules. However, once they're inside their host, they become active. Their basic structure consists of a protein shell surrounding nucleic acids. The nucleic acid inside them can either be DNA or RNA, but not both. It can come in the form of single-stranded DNA or double-stranded DNA. It could be single-stranded RNA, or something we don't often see, double-stranded RNA. Viruses are very specific to the host cells that they infect. So they have molecules on their surface that help them attach to the host cell. Again, they multiply by taking control of the host's genetic material and regulating the synthesis of new protein capsules and the nucleic acid core. They can't do any of their own metabolic processes, and they lack the machinery for synthesizing proteins. Now that we've had a good introduction, we'll move into our next section and take a look at the general structure of viruses.